Thank you for joining Plexon for part 3 of our 5-part webinar series featuring the NeuroLight optoelectrode. This webinar and the question and answer session will be available on the Plexon website following the live broadcast. Today's speaker, Lisette de la Prida from the Cajal Institute, will discuss how her lab is using the NeuroLight optoelectrode to probe hippocampal circuits. Thank you for joining us for her presentation titled Functional Microdissection of a Hippocampal Circuit with High-Density Optrodes. If you have questions for Lisette, please enter those questions in the Q&A section at any time. Please use the question and answer feature on Zoom rather than the chat feature for your questions. The speaker will address questions live during the question and answer session following the presentation.
Okay. Okay. Hi, hello. So thank you, Nayoka, for organizing this. I would like to thank UC and people from NeuroLight Technologies for the opportunity to work with the MicroLights for this talk. And thank you, Plexon team, for organizing this webinar. So along my talk, I will start first by posing the scientific question. In particularly, we are interested in understanding the organization of hippocampal circuit in health and disease. Then we will look for the appropriate tools to address this question. And we'll see how micro-LED optoelectrodes can allow for high-density recordings and control at very precise resolution. Finally, I will show examples and discuss approaches to gain enough resolution for dissecting the hippocampal microcircuits. But let's start with the problem. The problem is that the hippocampal circuit is organized along the proximo-distal and the radial axis. But what does it mean? This problem, as many others in neuroscience today, were proposed first by the Spanish Nobel Prize and the father of the field, Santiago Ramón y Cajal. Cajal drew some arrows in these beautiful drawings of brain regions, suggesting the direction of information flow running through the system. In this picture of the entorhinal of the hippocampal system of the hippocampus, we can see how cortical inputs perforate the fissure, enter the dentate gyrus, and establish synaptic contacts with granule cells, which are activated and send their axons to pyramidal cells in the CA3 region. CA3 pyramidal cells emit an axon collateral, which travels towards CA1 to activate CA1 pyramidal cells. Basically, over the time, we learned that there were additional organization in along these circuits. These 3 synaptic circuits that somehow simplify how information flow is proceeding through the hippocampus. We learned that pyramidal cells more proximal to the start of the circuit, they behave differently of those cells which were more located distally. This proximo-distal organization also applies to the CA1 region. And for instance, Schaffer collateral from the distal CA3 cells more like innervate proximal CA1 cells, whereas proximal CA3 pyramidal cells more like innervate the distal region. Such a proximo-distal organization is a major organizational axis in the hippocampus and play a critical role in the function. But along those early days, another researcher, Carolis Schaffer, also realized that there should be other arrows here. And in looking at how the CA3 collaterals, the Schaffer collaterals, run from CE3 to CE1, Schaffer also noted that the CE1 were comprised of large and small pyramidal cells, and that they were organized radially from the deep layers closer to the alveus to the more superficial layers closer to the stratum radiarum, which is a layer in the hippocampus CE1 where most of the parallel dendrites of pyramidal cells are organized. This organizational axis from the deep superficial correspond to the early lamination of the hippocampus. The hippocampus is, together with the entorhinal cortex, is considered the archicortex, and evolutively they appear earlier along evolution. And somehow this deep superficial lamination could be a primordium of the laminas that later are more elaborated in the neocortex. So somehow, if we want to understand the structure and the function of the hippocampus, we need to consider all these organization in terms of the proximal distal and the deep superficial axis. Now, among the many type of brain activities studied in neuroscience, probably the sharp wave ripples are one of the most well known, and this is the one of the most typical type of activity that you record in the hippocampus. It is originated in the hippocampus. It could be recorded when you isolate the hippocampus from the cortex, or even if you take slices of the hippocampus in vitro. And sharp wave ripple presumably are initiated in the CE3 region, and they travel proximal distal towards CE1. The sharp wave ripples can be maximally recorded around CE1. And for instance, if you use linear probe, high density silicon probe, which allow you to record local field potential from the different layers. And you can see that the sharp waves are typically maximal around the stratus radiarum. You collect these signals here, whereas the high frequency ripples are more located near the stratus pyramidale. To gain a more intracellular view of what single cells are doing during this event, we can insert a glass electrode within a cell. This is done in vivo under urethane anesthesia for stability purpose. And then we can see how the single cell fire during the events that are recorded in the CE1 with the silicon probe simultaneously. With the glass pipette, you can inject a dye, so you can, after recording, you can reconstruct the cell, validate the cell, and eventually evaluate the morphological specialities, like for instance, the thorny excrescences, which are typical of CE3 cells. During the recording, you can also use other criteria, like for instance, extracellular stimulation of the contralateral hippocampus to activate antidromically the projection of your CE3 cell interhemispheric, because this can give you some kind of blind validation that you are actually targeting the correct cell. When you look at how single CE3 cells are, the activity of single cells during sharp wave ripples across different membrane potential, you see them increasing the activity, depolarizing and firing just before, and this build up precedes somehow the sharp wave peak recording in the CE1. And this firing elaborates across the build-up and suggests that along these three synaptic circuits, these CE3 cells are projecting the firing toward the CE1. So based in the Schaffer collaterals and the arrows of we were discussing before, you expect that CE1 cells are basically depolarized, and you see them firing during the sharp wave ripples. However, some cells in the CE1 when recorded intracellularly under these conditions, they exhibit more complex interactions where you can see not only depolarization, but also depolarizing components. Morphologically recovery of this cell by looking at their location within the stratus pyramidale suggests that actually those cells which are more complex in their responses, having depolarization and hyperpolarization components, are located more far away from the border to radiatum, whereas those which are more likely to be depolarized are typically located closer to the border. And if you combine identification of the single cells here in green, one example, another example, with immunostaining, again, calbinding, a calcium chelator, you can see how cells more likely to be depolarized tend to be located closer to the border and positive for calbinding. However, those cells which are more farther away from the border, they do not express calbinding and they are located farther. This radial organization is related with this axis that I mentioned before that Schaffer suggested. Regarding deep cells, he is flipped. You see the basal dendrites and the apical dendrites. Deep CA1 pyramidal cells and superficial pyramidal cells, they born differently during the embryogenic development of the hippocampus. They express calbinding or do not express calbinding, and therefore they are two different cell types. Of course, there are always exceptions. You always have a deep cell which expresses calbinding or a superficial cell that do not express calbinding, but it basically calbinding and many other genetic markers confirms that there is this radial organization across the deep superficial layers in the rodent hippocampus. But to confirm that this functional segregation during Sharpeville ripples is not caused by the anesthesia that we have to use for the intracellular recordings, we use a manual microdrive developed by our colleague Robert Aberkin in Hungary. With this microdrive, you can advance a glass pipette together with an independent drive for the tungsten wire, and you can go to eventually establish a contact with the cell near the cell. But obtaining intracellular recording is quite difficult under this condition where the animal is not under anesthesia, but has many advantages as well. You cannot record membrane potential fluctuation, can bring the glass pipe close enough to see the cell firing, to stain and label the cell so that you can later recognize the cell and localize the cell within the layer. And you can also record by this with the very same glass pipette the extracellular field potentials nearby the cell. And that allows you to see whether under these drug-free conditions, there is also differences in the way in which the single pyramidal cells participate during Sharpeville ripple. Here, different cells, juxtacellular recordings in freely moving animals with these techniques are ranked according to their distance to the border with radialum. 0 here means that they are closer, so those are superficial, those are more deep cells. And as you can see, this is a firing rate histogram. Each row is a cell. There is an increase in firing around the 0 point, which is the peak of the shock wave. So this suggests that there is a gradient of participation during Sharpeville ripple from the superficial to the deep layers. And this gradient runs across few tens of microns in rats and mice. So, but if we want to understand how these differences emerge and how they contribute to the operation of the hippocampus and how hippocampus represent memory, episodic memory, and how episodic memory is built from the different attributes, we have to consider the modern view of the hippocampus today, in which we know how the different cell types in the entorhinal cortex project specifically by passing, in some cases, the 3-synaptic circuit that I was explaining before, and targeting different type of cells like intermediate inhibitory neurons that actually bring information from the different aspects like grids or border cells or cells coding for the speed, or even cells which are able to code for the time in between two events. And how all this is integrated into the hippocampal entorhinal circuit is extremely complex. And if we ever have the ability to understand how the hippocampus represents information, we require to take a more clear view of all these cell type heterogeneity. So that leads us to the problem. These very detailed approaches in terms of cell identity are not suitable enough to scale from the single cell to multiple cells. So the problem is posed, and we need somehow to be able to extract the information regarding the cell type specificity in terms, for instance, of whether the cell is calbinding positive or calbinding negative, regional specificity in terms of the tens of microns that I was mentioning before. So how to study the hippocampal organization at a large multicellular scale with enough specificity in terms of cell type and region specific, and that is the challenge. So that leads us to the tools and whether these micro-LED optoelectrodes can allow us to solve these problems. So the first who noted that the extracellular organization of hippocampal Sharpeville ripples can be used to extract specific information about the cellular components was Bushaki in this seminal paper by Mishuseki in 2011. Using high-density probe that were designed to perfectly match the rat hippocampal region, they noted that the ripple power reached a peak, a maximum, at some point across the shank. And they used that point to level 0 and to identify the location within the stratus pyramidale. Note also that the shape of the Sharp wave it also varies a lot across layers. We will get back to this point later in the talk. So they thought that basically by extracting information between how the action potential recording extracellularly from cells that were closer to a particular electrode but farther away from others, you can help combining with this to localize the location of the single unit activity across the shank. And that proved to work pretty well in terms of hopefully because the hippocampus is so well organized and the laminar organization is so straightforward that you can see cells which, for instance, are having their action potential, extracellular action potential amplitude being maximal more in electrodes which are more, for instance, closer to the border, to radiato, or more into these superficial regions, as compared with other units or cells that you see them firing and having their maximal action potential amplitudes in electrodes which are more located deep. And they are using this information to also try to infer whether there were a different functional organization across the deep and superficial layers. And in that paper, what they report was, for instance, the fact that by using these approaches to localize superficial and deep cells, they found that deep cells, but not superficial cells, they shifted their preferred theta phase firing position or timing during theta oscillation recording in a wave versus those recording during REM sleep. And they call this subset of deep cells that shifted the preference phases REM shifting cells. So somehow this tells you that in a way, using this high-density electrode can help to identify the position within the layer. But we wanted to validate how precise that could be, because as I was explaining before, this is a matter of maybe a couple of layers being superficial or deep, no more than 25 or 30 microns separation, because the C1 is quite packed. So what we took advantage of the fact that the juxtacellular pipette is close opposite over the cell And therefore, because we can record the extracellular ripple in this position, and we know exactly, because we recovered the cell position, we know exactly where we were recording the local field potential. And then we can extract whatever feature we like from the local field potential and look at how that feature in particular relate with the location of that particular recording from the border with the radiatum that we typically take as a reference. So here we see that for the different cells that we could label precisely, we can infer whatever feature of the event and plot that against the distance to radiatum. And if we couple that with the cal-binding immunostaining, we can precisely establish where is the limit between the deep and the superficial layer. Okay, so we examine many features of the shortwave ripple by filtering using different filters. Like each color here is the distance to radiatum, meaning that this ripple here, which is blue, was recorded closer to the border, and this ripple here, which is more red, was recorded far away from the border. These are in the supplementary figures of this paper. And as I said, we computed whatever measure of the extracellular event using different filters, and then we try to correlate that with the distance to radiatum and look for the best match possible. And we noted that the maximum ripple power basically peaked somehow a bit into the deep layers and not really at the border. And that the filtered low-pass filter 10 Hz shortwave event using information from the shape of the shortwave was informative also regarding. So we can use these too, but somehow, again, it's not quite easy to know whether you are deep or superficial. And basically, here is where the micro-LED optoelectrodes come in help. And we heard in the previous talk by Yusi Yoon 2 weeks ago about the design. These are 4 shanks. They come separated between one each other 250 microns. They are 70 microns width. And the location of the micro-LEDs are integrated into the probe so that you have the same layout of electrode I was describing before to localize across this and superficial. And now we have a particular micro-LED located precisely so that we can hopefully be able to activate and to look for the deep and superficial organization more precisely. In the last week's seminar, we learned from Missy Boros-Lakos from Bouchaki's lab how to use these electrodes, these probe, chronically. Today, I will show you how to use them using the head-fixed preparation instead. In the head-fixed prep, you can do acute recordings in awake mice, in rats, if you train them to stay and to run in a wheel or in a treadmill. And this is very convenient because this offers you the ability to use your stereotaxic frames on the apparatus that you already have in the lab to place the probe and to bring the probes within the brain to your favorite target area. And basically, the probe comes with 2 magnetic channels, magnetic connectors. One magnetic connector allows you to plug the probe to the head stage for recording the electrophysiological signals. And in the back of the probe, you have another smaller connector for powering the LED. What we typically do is, well, you need to solve the probe for the acute preparation. You need to solve the one connector for the ground and one connector for the reference. And then we typically use intent head stages. And what we do is that we solve a metal bar to the head stage ground in the PCB. And then we use the fixation system to hold the head stage this way, and therefore the whole preparation is grounded. Then with the ground connector, you can connect to your head plates that you have in the animal, implanted in the animal. and then the preparation is ready for recording. Basically, this is the electronic circuit connection, which basically, as I said, you have the electronic part for recording the signals, and then you have the other connector going through the controller for powering the LIDs. And here we use the current controller developed by NeuroLight technologies for the purpose of controlling the probe. So the head fixed preparation has many advantages. It is easy to use and very easy to troubleshoot if you have any problem. It's more simple to learn, particularly if you don't have experience in chronic recordings. It allows you evaluating basal physiology because you can have your animal doing nothing really, or just, I mean, they like to be, well, they get a bit habituated to be in the wheel, and they can run, they can stop, they spend the time over there. Or you can couple that with cognitive tasks, I will show you later. In this probe, you can readjust craniotomies, and therefore you can somehow improve your targeting your coordinates. So something which is I found very, very interesting in this preparation and very important is that it allows you to do several sessions per animal. And by sessions, I mean the penetration track. You just got chronically, you can go deep or not, but you cannot move in the proximal-distal or in the another coordinate, and that gives you very strong and more power, statistical power, to reduce the number of animals that you need while reaching a robust statistical conclusion. And finally, another thing which is a great advantage is that you can reuse the probe several times. For instance, in this probe in particular was used in 6 mice. We use them between 3 and 4 sessions per mice. And you can use enzymatic solutions like this one, but you can look for any other. We use this tergasim solution at 2%, but as I said, I mean, it's not the only one. And here you can see before and after the same probe. After we leave the probe immersed, submerged all the night in this enzymatic solution, and the day after you clean the probe, and you have the probe perfectly for new penetrations. So, as I said, the head fixed prep can be coupled with virtual reality to develop different tasks. For instance, this is one of our setups. I don't know if you can see this small movement. Basically, is simulating the fact that when the animal moves the wheel, they run across a virtual corridor, and then you can couple that with whatever combination of stimuli you want, and you can design your own tasks, and the animal learn to do that, and then you can couple that with the recording with the probe using, as I said, your micro manipulator. You can use reinforcement for having the animal, or even you can couple that with sounds and design whatever combination of stimuli you want to have the spatial mapping that you want to address in the region of interest. And inserting the probe with an appropriate orientation into the animal, into the brain, allows you to obtain radial recordings. Here are recordings in the hippocampus from each shank of the 4 shanks is penetrating the CA1 dorsal region from the deep to the superficial. Remember, in the hippocampus, deep is coming first, is flipped as compared to the cortex in the dorsal region. So first you see deep layers, sublayers, and then you go to more superficial layers, and you, as you can see, you can record action potentials everywhere from the proximal to the distal CA1 region. That gives you a lot of information. Note, for instance, how the chart wave shape can provide precise information regarding location within each of the channels. Okay. So, equipped with these tools, we can now think that great. We have a tool to scale from single cells up to multiple cells. But how precise can we activate deep and superficial CA1 cells in a given shank? This is a big question, as I spent the first part of my talk giving you a lot of information using single cell approaches, how precise we can be with single cell approaches by juxtacellular or intracellular approaches regarding the location of the cell. And I also spent some time in showing you how challenging is the fact that we have this deep superficial organization really packed in the CA1 region. And it's just a matter of tens of microns differences from the most superficial layer to the most deep layer. Probably this is one of the most challenging dissection that you are facing. Typically what you do for optogenetic stimulation is you use some adenovirus and express the opsin, for instance, channel rhodopsin, under the control of a particular promoter, in this case CAMK2, which allow expression of channel rhodopsin specifically in pyramidal cells in a wild-type context. But as you can see, then you have fully coverage of the opsin expression across the deep and the superficial layers. So you need to penetrate the region very slowly to locate the probe across the region and to use your stereotaxic information and learn from your spontaneous events to know precisely where you are, to infer precisely where you are. Once you are properly located, the hope is that with this micro-LED design, it will allow you to precisely control deep and superficial cells separately. But remember that deep and superficial CA1 pyramidal cells are really packed and that they are just sometimes 20, 30, 40 microns apart. So it's really challenging. With a probe, it works pretty well in, as I said, in localizing the probe across the layer. If you learn from your favorite event, like the ripples for the hippocampus, that whether you are more in the deep and the superficial, and then you can isolate single spike from individual units. For instance, this unit here, the maximal amplitude of the action potential is in this channel 4, 1, 2, 3, 4, this channel here. And therefore, you can think that this cell is more or less located intermediate in the layer. Now, shining the light in one LED, in one shank, should be pretty specific, and actually it is the case. As you can see, you can shine light in one of the shank, for instance, the shank number 4, and you see the cells single unit firing in shank number 4, but not really responses in the other shank number 3, which is 250 microns apart. These are the artifacts of stimulation artifacts that you can get rid of with the new generation probe that Yusik was mentioning before in his talk a couple of weeks ago. We like artifacts, I have to say. I mean, it's good not to have them, but for us, for instance, they are very useful to facilitate identifying where the stimulation was applied. Now, using the probes, we can clearly identify individual units. In this example that I was showing before, you see how by shining the light of the micro-LED, which was more proximal to the sites where we record the maximal amplitude action potential, we saw the single cell firing. We increased the firing. Here is the different trials that we apply stimulation, like 15 in this case, and you see that just systematically each tick here is an action potential. Without the stimulation, you see the background potential and the firing rate, and when you stimulate, you see an increase that actually you can estimate a kind of firing rate curve that tell you how much you are activating the cell. Now, in the dorsal CA1, connectivity between pyramidal cells is extremely rare, and this makes our lives a bit easier. So we expect that if we saw pyramidal cells responding to light here, is because they are expressing the opsin. We are somehow discarding, by connectivity between pyramidal cells very low, but we are somehow discarding the indirect effect. However, if you are working in different structures, you need to learn how these indirect effects are shaping the response. But in the hippocampus, for instance, something that is strongly connected is pyramidal cell to interneurons. These feedback inhibitory loops are very important in the organization of the hippocampus. And actually, you can see some cells that when you when you illuminate one LED, they decrease firing, suggesting that they were inhibited by an interposed interneurons that were activated by the LED. So in trying to understand, to evaluate these type of changes, we can estimate a firing rate gain by using information set scoring the basal firing rate of the cell, and then we can evaluate whether the response was increased, therefore excitatory response, increasing firing or decreasing firing, and somehow suppressing firing here. And then basically you represent information in a matrix. In this matrix, what you see here is here are the channels, so 1, 2, 3, 4, 5, 6, and so on till 8. So this is located superficial in the CA1, and this is deep. And the micro LED from number 3, 2, and 1 from the deep to the superficial. So in a row here, what you see is that for all the units that you were located firing, and as you can see, for the LED number 3, things to be quite specific in these experiments. So we shed light, and we see responses in channel 1, 2, 3, 4, and 5, and then we see no clear differences. So that seems to be quite organized. However, in this case, as you see, if you shed light by the LED number 2, you have a mixture of responses. This is because the obscene is not expressed sparsely. It's expressed in all the layer, and not only at the soma level. They are also expressed in the basal dendrites, and therefore, even if the cell is located here, you may have a basal dendrite going through this way, and therefore you activate the obscene. So, although you can get some specificity, we need to twist the tool a bit more to improve it. And this is what we are doing actually by coupling the micro LEDs with cell type specific optogenetics. So we use a cal-binding Cree animal that allow us to express channel rhodopsin in the Cree context under the cal-binding positive cells, and we express and then we can confirm specificity by looking at the percentage of cells expressing our reporter coming with the channel rhodopsin, suggesting that expression is quite specific in the superficial layers. For deep cells, we use a type 1 line, which allow expression mostly in deep cells compared with superficial, and this together coupled with the micro LED allow now us to be more precise in terms of how we are activating the different cells. This is one example. Again, 1, 2, 3, 4, 8, more deep superficial layers, and as you can see, we can shed light and activate single cells in the superficial, in the cal-binding animal, and using the type 1, we can be more precise in activating the deep. And actually, this is how the response matrix now looks like for the type 1. This was the previous one in which we were having on a specific expression of channel rhodopsin all along the layer, and as we were discussing, it was not so clear whether we can control deep and superficial layers efficiently, but in these two other cases, as you see, we gain the specificity we wanted by recording, by activating more precisely. You see green here is responses by the micro LED number 3, in the deep layers in the type 1 context, and the opposite for the cal-binding animal, having more responses here by activating the micro LED number 1 and 2, and having responses more in units that were located more deep in the. So great tools now we can move on and have a look to the data. And I will only show you some glimpses of our data to illustrate how you can use these tools and how these can allow you to separate and segregate the activity, and how this is segregated along the radial axis in the C1. One thing that is good about the tool is that now we can do control activation of multiple units, deep and superficial, very precisely. This is one example in the calbinding tree. Here I'm showing the amount of currents we are applying to one particular micro LED, and here is a measure of the LFP power. And as you see, the more we apply, the larger the response is. You will see how in the next examples. So if I am moving into the low current between 10 and 30 microamperes, they can control whether we shed more or less light. That will depend, of course, of the level of opsin expression that you have, and precisely activate fuse cells. These fuse cells that are activated during light, you also see some of them firing spontaneously when you are not applying the stimulation. And then you can use your favorite sorting program to evaluate which units were automatic sorting of all the 8 channels per shank, followed by 5 for visualization and manual correction. And then so we know exactly the location of the cell within the shank. We look at the spike waveform of the cell and look at the firing dynamic of this cell, the spontaneous dynamic, by using the autocorrelograms. Those are criteria that then you can use combining with sorting to separate pyramidal cells from putative interneuron. We also have information about collect information about where the cell is firing along theta, whether firing in the falling phase of theta in these particular cells, whether the cell. We can also track the participation of the cell during sharp wave ripples, and using information of the ripple power or of the sharp wave shape of the filtered traces, as I explained before. Running in a wheel, as I show you, allows you to have to extract spatial information. In this particular case, the cell was not having any spatial tuning, but we can also calculate phase precession and other features regarding the spatial representation and other kinematic properties, like, for instance, whether the cell is speed modulated or not. So we can putatively identify different cells that we can somehow classify as deep or not, depending on their location in the track, using the information as was explaining before, and then try to see whether these cells were selectivity activated or not. In this particular example, you see each, in this matrix, we have each of the cells units, putative units that were classified, like, for instance, deep pyramidal cell, this another deep, some sup. We can even find some cell that we can classify as fast spiking paravolum positive basket cell interneurons, and so on. And here we see how the different cells that were recorded together in the same experiments and were responding to the micro LED specifically, whether we shed the light in the shank 1, number 2, 3, or 4 across the micro LED 3, 2, or 1. And for instance, you see this specificity that the cell is responding more vigorously. Here is activation is red, inhibition is blue. When you shed light, for instance, from the more deep LED as compared with the more superficial LED in the shank where the cell is seated. But also you see that there are interactions, and there is poor interaction between the channels as you move across them. Using this information, we can somehow map the circuit more specifically and try to identify the specific connectivity between deep and superficial cells with different type of interneurons. Another interesting application here is optotagging multiple units, and for instance, here using the approach, we can record or identify cells which are prototypically deep cells, optotagged as deep or superficial, and evaluate, for instance, whether the deep cells were firing in a particular theta phase, and for instance, mostly firing near the theta peak and in the early falling phase of the theta, recording in the stratus pyramidale, whereas superficial, optotagged superficial galvanizing positive cell, they're more likely to fire near the theta trough, recorded in the stratus pyramidale, and even compare that from the optoelectrodes with the single cell juxtacellular recording, in the awake head fixed condition, or in the freely moving condition in the rat, and validate across animals, across preparations, across strains. We can also look at the connectivity across, across the proximal distal axis. For instance, in the previous matrix, that you know that some cells are more likely to be inhibited. Like for instance, this actually is interesting because some parallel positive cell could be activated within a sham because they are probably feed-forward activated or feedback activated by some interposed pyramidal cells. But also they are somehow inhibited across the shams, and that can give you a lot of information on how there is the proximodistal organization of the feedback inhibition, which can be evaluated more precisely in different mouse lines. Here in these matrices, for instance, we have the responses here. Activation is green, inhibition is red, as I showed before. The channel 0, and then we look at within the channel how is the mean response across all the channels. When we shed light in the number 3, 2, and 1, here is wrong number, no problem. And you see that when you shed light in this channel, in the next chance, 250 microns apart, actually you see more inhibition than excitation, and there is an asymmetry in the way this is organized for the type 1 or the cal binding, or even if you use the non-specific expression. Then if you you can shed more light and using the very same sham that before we see specificity on the single cell that we were activating. If we increase the power of the light, we can actually induce ripples. We can induce synthetic local field potential oscillations in the range of the frequency range of ripples, and these synthetic ripples actually could be differently have different properties depending on whether we activate them by activating the superficial or the deep cells, and we can use these to compare the synthetic ripples with the spontaneous ripple, for instance, recording in the same channel. So if you like to learn more about these, many things that you can do with these tools, you can read some previous papers that describe the technology and using these to extract information. And I'm coming to the next webinars in the series by Dan English and by Sam McKinsey. So I hope that you have seen how by coupling micro-LED technology with cell type specific optogenetics, you were able to more precise mapping of activity, particularly regarding, particularly regarding the sublayer specificity of the hippocampal function. I take the opportunity to bring the attention to the Google groups that can help for community sharing and looking for answers and trying to troubleshoot. And before to finish, I would like to acknowledge you for your attention and the funding agency, but most especially my team in Madrid, especially Teresa and Alberto and Elena for experimental part of the work, and Andrea and Kike for the analysis. Special thanks to UC June for facilitating us the access to the technology, and Robert, Aberking, and Manu Valero for the single cell approaches, which were the experiments that opened the questions in the lab for trying to understand this deep superficial organization of the hippocampus. So thank you for your attention. I'm happy to take questions.